Hi, I'm Susanna with Science with Susanna, and in this video, we're going to finish going through the chemistry foundations that anatomy and physiology students will need for success in their coming studies. In mixtures of atoms, each of the components actually retains its own properties because it's not a chemical reaction. So when we have mixtures of atoms and molecules together, each component retains its identity. Whoop, there. And I'm going to show you three kinds. There are suspensions of mixtures. There are colloids. And probably the best known are the solutions. There are suspensions, colloids, and solutions. A good example of a suspension is your blood. A good example of a colloid is homogenized milk, which is what most people buy at the store. So, milk from your fridge. And an example of a solution is sugar water, like Kool-Aid. Let's draw a little test tube here to compare blood, milk, and sugar water. So, these are all different kinds of mixtures that exist in the human body. And as I explain these to you, you can think of the example of the blood as the least well mixed, and then milk is somewhere in the middle, and then the sugar water would be the most mixed, most uniform. For the blood sample, when blood sits for a little bit, if it's not flowing, the red blood cells settle out at the bottom, and then at the top, you have the watery portion of blood, which does contain all kinds of things like electrolytes and hormones, but the bottom part is where the red blood cells settle out. A suspension is having large particles that will settle out. Then, if we look at the solution, they're going to have extremely small particles. So, they're going to have the smallest particles, and it shouldn't settle out. Water will have dissolved the other components. In my example that I gave, it would be like sugar. So, you would have in the water, you would have the sugar molecules totally and uniformly surrounded by the water molecules. Remember that water is good at doing that because it is a polar molecule and sugar is also polar. And so water is going to be able to dissolve the polar sugar or glucose molecules. Homogenized milk is our third example. You're going to have some somewhat large milk proteins and milk fat molecules that are spread throughout the mixture. The particles are a lot larger than, for example, the glucose we discussed. We'll just say these are mid-sized particles, and they won't settle out. As we study anatomy and physiology through this course, we will be considering different types of mixtures in the body and then the characteristics they have based on how well mixed they are. Chemical reactions are different than mixtures. In a chemical reaction, you have new bonds that are formed and broken. in order to create a new compound. So we're going to see covalent or ionic bonds either formed or broken. And some chemical reactions are more complicated than others, and so you have some bonds formed, some bonds broken in the same chemical reaction. First, let's look at an example of a chemical reaction where something is built, where we're going to form a new bond. Let's say you have molecule A and you have molecule B, and then these can be put together to form a new compound called AB. A covalent bond was formed between those. This is how you write a chemical reaction, and these over here are called the reactants. Now, when there is an enzyme involved, that's something that will help to speed up the chemical reaction. We call that the substrates when an enzyme is involved, which in human body, pretty much always enzymes are involved. And then what gets formed is going to be called the product. In our body, when you have a chemical reaction where something got built, we call it anabolic, where you have smaller molecules put together to build something bigger. So something gets built or synthesized. So we take smaller things and make something bigger. Now, these type of anabolic reactions in our body usually are what we call endergonic. That means that they require energy in order to proceed. I can draw you a little picture to show you how this works in the body. Let's draw a little graph like this, put time here and energy here, and we'll use a blue line to represent the reactants right here, okay? So these are going to be the reactants or the substrates, and they have a certain amount of energy, right? Then the reaction proceeds, and you have to put in energy called activation energy to get it started. But then once it gets going, it will come back down a little bit, and the products then are right here and represent a higher amount of energy. So if I use this line right here, do you see how you actually have products that have more energy than the original reactants did? So we can draw a line right here and show that all of this was the energy that was required. Energy added. Now, let's take that same product and think about in our body, maybe when we need to digest something and we want to break it down, then the reactants now would be the AB. There's just one reactant then. And with enzymes involved, we're going to call this the substrate the enzyme can act on. And then the reaction will proceed, and the covalent bonds between those two molecules will be broken, and we separate them out into the products A and B. This kind of a reaction we say is catabolic or breaking. So it's going to take bigger molecules and break them down into smaller molecules. And sometimes you'll see this called decomposed, breaks down. So it's really important for you and your foundation with anatomy and physiology to know that anabolic are reactions that build things in your body. For example, building muscle, when we put that here. When you work out and you're making new muscle proteins, this would be considered anabolic. And when you're eating, then a good example would be digesting your food. And these catabolic reactions usually release energy. And so we say that they are exergonic. And I can show you that on our little graph too. So let's use a green line and we're going to start with those products, the AB. So it's like right here, right? You're still going to need activation energy. But once that reaction gets going, now energy is released. And so the products right here, this would be A plus B, and the reactants were A, B, whereas it was the other way around for the endergonic reaction. You had A plus B go together to make A, B, like that. Energy released. I've thrown around this word enzymes now a little bit, but what is an enzyme and how does it work? So, enzymes are catalysts that speed up chemical reactions. Enzymes are biological catalysts, and a catalyst is something that speeds up a chemical reaction. And an enzyme is a special word for it in biology because it's usually a carbon compound. I'll use this graph again to show you how an enzyme can be helpful in a biological chemical reaction. In the human body, we need enzymes for virtually all of our chemical reactions. And without them, the reactions would happen so slowly that we would be dead before we could digest our food. So here we'll put time on the bottom right here and energy on the side like we did before. And I'm going to show you with and without an enzyme what it would look like. Let's use that blue line like we did before. And remember, we had reactants, let's say with this amount of energy, and then we ended up here like that. That I was showing you an endergonic reaction. So we could put the same of this on both graphs, okay? So you've got the energy of the reactants and then the energy of the products. You can put maybe an R and a P, an R and a P. Now, without an enzyme, the activation energy might look like this, okay? So this difference right here is the activation energy. Now, when you have an enzyme at play, look at what can happen. Do you see that? Now the activation energy is less. So we could say that number one, enzymes are biological catalysts. And a second thing to say about them is they speed up chemical reactions by lowering activation energy. And make it easier for the reaction to proceed. Another thing to remember about enzymes is that they are usually proteins, not always, but usually, and their shape makes them very specific for their particular substrates. And those are going to be the molecules that they bind to and help the reaction to proceed. And remember, the substrates are very similar to the word reactants. A cool thing about enzymes is that they can be reused over and over again. They don't get changed during the reaction. Because enzymes are protein and their shape is so important to their function, they can be ruined if they are put in an acidic environment or too hot of an environment. So we'll use the word denatured here. So they don't work anymore. They get unfolded. And then when they don't have the right shape, they can't act as an enzyme. And we'll talk about this more, but basically, think about pH or boiling temperatures are two things that are very well known for doing this. On this page, I'll describe enzyme structure and function. I'm going to start by drawing an enzyme right here. They're really big proteins. And I'm going to put a couple of spots on it that we'll call active sites. I'm going to color this enzyme blue, and then we can put a label on it. And these spots that you see right here and here, these are called the active sites of the enzyme. That's where the substrates bind. And what are the substrates? Let's look at that equation again that we had. We had molecule A plus molecule B have a covalent bond form that between them to form molecule AB. And in this case, remember these were the reactants or the substrates. And then here's the product. Let's represent molecule A with a purple circle and molecule B, we could just make like a red triangle. So these are two different molecules, and they are going to be brought together to form a new product. Maybe the new product will look something like this. So the enzyme works by binding to the substrates. And in order to do this, it actually changes shape a little bit to fit them better. I don't know how good that looks, but so you're going to have the purple A molecule can fit in right here. And then that red triangle can fit in right here like this. And you see how now they're brought closer together? And by being brought closer together, it makes it easier for them to have a bond formed between them. So the enzyme is facilitating this reaction. It lowers the activation energy. It gives them a place to sit. You can have a covalent bond formed. And remember, I told you that enzymes can be reused. So once it has facilitated this chemical reaction, it goes back to its original shape and it's ready to be reused again. And then it goes back to its original shape and it's ready to be reused. So this was an anabolic reaction, right? It was building something bigger. And now Let's look at a catabolic reaction example. Because almost everything that happens in your body needs enzymes. In fact, usually multiple enzymes just to get one thing accomplished. So now let's draw a green enzyme. This is going to be a different enzyme. Remember, they are highly specific. So this one is going to be designed so that it can hold onto the bigger molecule and break it down into the smaller molecules. In our catabolic reaction, we're going to start with the substrate, AB, and with the help of the enzyme and the lowering of activation energy, we're going to break that apart. We're going to break the covalent bond. In this case, the enzyme, so it's going to pop the two apart, facilitating the breaking of a covalent bond. But like the previous situation, the enzyme can go right back to its original shape and it is ready to interact with another substrate. So we could say that in both cases, these are ready to be reused. Let's put the word enzyme on here. This is going to be a different enzyme in green than we have in blue up above. So remember that enzymes are highly specific for their substrate. They work by lowering activation energy, and they can be denatured by either really high or really low pH or by boiling, and this will ruin it. This is why if someone gets a very high fever for a long period of time, or even not such a long period of time, their enzymes will denature and their brain will stop working and they will die. The pH scale is a way for us to talk in biology about how acidic or basic different parts of our body or different secretions are. The pH scale goes from 0 all the way to 14, and it's a logarithmic scale. So each number is going to be 10 times more than the one before it. The middle number is 7, and that is going to be neutral substances. And that what that means is that their concentration of hydrogen ions is going to be equal to their concentration of hydroxyl ions. And really, the only substance that's going to fit that description is going to be pure water. So, pure water is neutral and has the same amount of hydrogen ions as it does hydroxyls. Now, everything that has more hydrogen ions than that is considered acidic. And I'm going to color this, everything down here, a red pink. So this is acidic, everything less than 7, and everything greater than 7, all the way up to 14, is more and more basic. So these are all going to be basic, or we use the word alkaline. Everything less than 7 is acidic. We measure it by the amount of hydrogen ions. So the most hydrogen ions are going to be at the very most. So hydrogen ions increase with increasing acidity. A good example of a very acidic thing in our body is stomach acid. Stomach acid has a very low pH. It's only 2, which means that it has a lot of hydrogen ions. And hydrochloric acid is the substance that makes up stomach acid. And when it is mixed with water, it will donate a hydrogen and then separate into its ion form like that. So we can define acids as substances that donate hydrogens. They donate, or honestly, I usually would say they give up hydrogen ions in water. We can contrast that with bases. Bases are going to be able to accept, or again, I use the phrase pick up hydrogen ions. This is what matters to me because we need to make sure that our blood does not get too acidic or too basic. Speaking of blood, the pH of blood is very tightly controlled in your body between 7.35 and 7.45. So it's a tiny bit basic. If blood is higher than this, the patient is considered to be experiencing alkalosis. And if the blood pH is anything less than this, we say that the patient is experiencing acidosis. To give a good example of a substance that is a little bit alkaline in the body to contrast that very low pH of stomach acid, we can look at bicarbonate, which has a pH of about 8. And bicarbonate, it's like baking soda basically, is secreted by your pancreas, and it goes into the small intestine, and then it neutralizes the stomach acid. So stomach acid doesn't burn your small intestine. Recall that enzymes only work when they're at the proper pH and the proper shape. This is also critical for digestive enzymes to work right. It's so important to keep our bodily fluids at the right pH that we have many buffers in our body naturally. And buffers are substances that are able to pick up or donate hydrogens as they pass through fluids. So if there are too many hydrogen ions around, they can just mop them up. And if there are not enough hydrogens around, then they can just drop off some hydrogens. So they can resist pH changes to keep us healthy, maintain homeostasis. Picking. Or so they can do whatever's needed. To repeat that, if there are too many hydrogens, they can bond to those and take them out of the water. And if there's not enough hydrogens, they can drop some off. Important buffers that we have in our body are hemoglobin, which is a protein that carries oxygen. So we have a lot of it, and it's always around in the blood and able to then act as a buffer. And the other example, let's get rid of this. The other example is called carbonic acid. And this is the most important buffer in our blood. So, two examples of buffers: carbonic acid and hemoglobin. These are examples of buffers in blood. So, not only do we have substances in our blood and bodily fluids that act like buffers to resist changes, but we have two body systems that are very focused on balancing our pH. Those include the urinary system and the respiratory system. These both work hard to balance our pH. So, respiratory system, when we breathe, we can blow off carbon dioxide, which makes our blood acidic. So, that's what our respiratory system can do. And then our urinary system can pee out excess hydrogens or retain them if needed. So we have two body systems that are very good at helping to make sure we keep our pH in balance. We are on to macromolecules. We are really coming into the home stretch of building your chemistry foundation for anatomy and physiology. This is the last topic we'll need to go over. So, macromolecules are huge. They are large biological compounds based on carbon, and they are what make up your body. They also do all the work inside of you. There are four different kinds of macromolecules that make up all the different organic parts of you. And when I say organic, I mean formed by carbon. First are carbohydrates. Put a box around those. Next, we'll talk about lipids, then proteins, and finally, nucleic acids. The foods that we eat contain all four of these in different combinations. Carbohydrates are built from simple sugars. Lipids are built from fatty acids. Proteins are built from amino acids. And nucleic acids are built from nucleotides. We spent a lot of time in the previous video going over the periodic table and important elements. And that's how we are going to classify these. So they're all going to contain carbon, right? Because they are all organic molecules, which by definition means they have carbon in them. And they also have hydrogen and oxygen in them. Remember, I taught you that CHON, carbon, hydrogen, oxygen, and nitrogen, are the most common elements that make up most of your mass. And that's because they're in these macromolecules, and these are what form you. Lipids also have carbon, hydrogen, and oxygen. And sometimes they have phosphorus. Proteins also have carbon, hydrogen, oxygen, and each of them will always have a nitrogen. And some proteins have sulfur. Nucleic acids are really big, so they're going to have carbon, hydrogen, oxygen, nitrogen, and phosphorus. Every single one of them will contain that. Now I'll try to show you an example of each type. Let's take a simple sugar like glucose, and it would have this formula. like this. See if you can color along with me. It's not so bad. Just we make these little lines to represent bonds. See that? How it makes like a ring structure? And then coming off of this top carbon, there is yet one more carbon that's not in the ring, but part of the molecule. And all these carbons have little OH groups or hydrogens coming off of them so that carbon gets to form its four bonds like it always wants to do. See how there's so many oxygens? And that's going to make this a polar molecule. I'm going to use my yellow and look at all of these oxygens. You see all these? Because it's a pretty small monomer, this little glucose, and it has all those oxygens, this is going to make carbohydrates hydrophilic, meaning that they are polar, and it means that they're going to dissolve easily in water. Let's contrast that with lipids that tend to be hydrophobic, and here's why. They have a carbon with an oxygen double bonded, and this is called a carboxyl group right here. They have this, right? So you think, oh, I see a couple oxygens. They must be polar. But no, because look, they have this long carbon chain. This is called a fatty acid tail. And this thing can go on for 16 carbons. And then each of these would have hydrogens attached like this. And if it has as many hydrogens attached as possible, then we say it's saturated. And if it has some places where it has double bonds on a couple of the carbons, then we call it unsaturated. And I'll show you that briefly on a future page here. But because there's actually only a couple of oxygens at one end of the molecule, then lipids tend to be hydrophobic or nonpolar and don't dissolve easily. So that's different than a carbohydrate, a big difference. Okay, then we'll look at proteins and an example of what one amino acid would look like. So they're going to have a nitrogen, the end. This is called an amine group. This is how they get called an amino acid. It's where their name comes from. And then there's a central carbon and then a carboxyl group on the end. But there's this R group right here. The R group is unique for each amino acid. It's going to determine whether or not the amino acid is hydrophobic or hydrophilic. So if the R group has a bunch of oxygens in it, then yeah, this protein will be hydrophilic. If it doesn't have very many oxygens in it, then it's going to be hydrophobic. So they can be either, and it comes down to what atoms make up that R group. There's 20 different amino acids. Nucleic acids are pretty firmly hydrophilic, and that's because they have a phosphate group with a bunch of oxygens on it. So it's very hydrophilic. And then they have a sugar that has, like we looked at the glucose, so it's similar to that. So it's also hydrophilic. And then they have what's called a nitrogen base. You hear the nitrogen in there. Nitrogens are electronegative atoms, and so they're going to make it hydrophilic too. So nucleic acids are strongly hydrophilic. Now I'll discuss carbohydrates a little bit more. Carbohydrates provide our bodies with quick energy, good fuel, and they're important in identifying our cells along with proteins. The building block of carbohydrates are called saccharides. This means one sugar. And I'm just going to represent it with a red circle. Examples of monosaccharides are glucose. This is what is in our blood when we talk about blood glucose. Then there's fructose. This is a fruit sugar, and galactose. These are the three best known simple sugars. They can be put together to form disaccharides. And disaccharides means two sugars. So if you put together two glucose molecules or a glucose and a fructose and connect them with a covalent bond, then you can make something like sucrose, which is table sugar, or lactose, which is milk sugar. And if many more are put together like this with covalent bonds linking them, you can form polysaccharides. And polysaccharides means many sugars. So I'll use a bunch of reds to show that now we've linked together many simple sugars to form starch, for example. Dietary fiber is a polysaccharide, and so is glycogen that we store in our muscles for quick energy. The building of these polysaccharides is considered an anabolic process, so it's building, and it uses dehydration synthesis. In the process of dehydration synthesis, covalent bonds are formed between the various sugars, and in that process, water is removed. Water is removed to form covalent bonds. Breaking down polysaccharides is a catabolic process. And it uses a process called hydrolysis, which is the opposite process from dehydration synthesis. Hydrolysis means water break. So we have to add back water in order to break the covalent bonds and break down the polysaccharide into simple sugars, for example, when we're digesting. So we're adding back that water in order to break the bonds. In anatomy and physiology, you'll be learning about a lot of different enzymes, so it helps to get familiar with their names early on. Amylase is an enzyme that can break polysaccharides or starch down into simpler sugars. We make amylase, this enzyme, and produce it both in our saliva and from our pancreas. Disaccharides like sucrose, lactose, and another one known as maltose are broken down by additional enzymes such as sucrase, lactase, and maltase. Notice that enzymes often end in ase, and so these enzymes are going to break down those disaccharides. Next, we'll discuss lipids. So we talked about carbohydrates, now we're talking about lipids. They serve to provide long-term energy for our body. They make up your cell membranes. They are the foundation for steroid hormones. They form myelin, which is a fatty insulation on your nerves. And they insulate and cushion your body. Lipids in the human body can be classified into three main groupings. First, the triglycerides. You can see right here, they're going to have three fatty acid tails. Then the phospholipids. These are the ones that make up our cell membranes. And lastly, the steroids. The structure of a triglyceride is that it has a glycerol head, and that has some carbons, oxygens, hydrogens in it. And then it has three fatty acid tails, like this. One, two, three fatty acid tails. Fatty acid tails can be either what we call saturated or unsaturated. What I've drawn here are all saturated fatty acid tails because they're all composed of single bonds. But if I had made one with a double bond, this, then we would call this a mono unsaturated. And this ends up making the fatty acid not be able to bond to as many hydrogens because the carbon is having two bonds with another carbon. And that's why it's unsaturated because it can't hold as many hydrogens because it's taking up a bond, an extra bond with another carbon. Now, if the fatty acid had two or more double bonds, like this one, then we call this polyunsaturated. DB is for double bonds. The primary job of triglycerides in our body is as energy storage. There is a lot of energy trapped in those carbon bonds. So we can store that energy in our adipose tissue. And we have an enzyme called lipase that helps to break apart these carbon chains. Your pancreas makes this enzyme called lipase, and it's important in the digestive process. Triglycerides are also going to cushion organs and insulate and keep them warm. Next are the phospholipids. So instead of a glycerol head, phospholipids have a phosphate head. And this makes this end of them actually pretty hydrophilic. And that's important in the structure that they make up, which are your cell membranes. So they form cell membranes and they form what's called a lipid bilayer. They also have fatty acid tails, but instead of having three, they only have 2. So 1, 2. And like the triglycerides, the fatty acids may or may not have double bonds in them. There's a lot of different kinds. The third category of lipids in the body are steroids. Steroids are hormones that are formed from cholesterol. Not all hormones are steroids, but some very important ones in your body are. So the structure of a steroid is much different than what we've looked at with the other lipids. Look at this. You're going to make 3 rings like this. They call this like a sterol ring, like that. And then make a 5-carbon ring right here. And then just go ahead and put a little tail off the end. This is a little cartoon of cholesterol. And cholesterol then can be modified in various ways to produce the following important steroid hormones. Vitamin D is a steroid hormone. Testosterone. Can you guess another one then? Estrogen. Can you guess another one then? Progesterone. And these are the sex hormones then. Your adrenal gland can make cortisol. This is what some people call the stress hormone, and aldosterone, which also is associated with handling stress and making sure your blood pressure stays adequately high and things like that. We've talked about carbohydrates, lipids, and now we'll talk about proteins. And this one needs a whole page just to tell you its functions. It has so many functions in our body. Many structural roles of protein. Proteins that maybe you've heard of include keratin and collagen. Collagen is actually the most plentiful protein in your body. These proteins make up parts of you, such as your hair, your skin, and your nails. Collagen fibers also form your tendons and your ligaments. Proteins also function in movement. Actin and myosin are 2 contractile proteins that are found in your muscles. Muscle fibers are heavily composed of these proteins, actin and call them contractile proteins because they can shorten. Isn't that awesome? Then there's defense. Your antibodies are proteins. Another word for antibody is immunoglobulin. And we almost always draw antibodies in this Y shape. Most enzymes are proteins. You'll be learning about enzymes every chapter when you study anatomy and physiology. Just a few enzymes that I've already talked about include lactase, amylase, lipase, and I can't remember if I mentioned pepsin, but it starts to break down proteins. All of these are examples of enzymes, and they're all proteins. Then transport. Proteins like hemoglobin transport oxygen, or transferrin transport iron, or lipoproteins transport cholesterol and other lipids. And then one last role is antigens. These are identifying flags on cells. These are usually what we call glycoproteins, meaning that they have carbohydrate and protein. We can draw a cell, this, and then it might have a glycoprotein on its surface that acts to identify, oh, I'm a liver cell, or oh, I'm a muscle cell. Now that I've explained a few of protein's functions, let's talk about how proteins are built. Protein building. Like carbohydrates and lipids, proteins have a monomer, and theirs is called amino acids. And like I did on the other pages, I'll use a red dot to represent just this would be like one amino acid. These amino acids can be put together to form dipeptides. Di means two. So this would be two amino acids that are linked together with a covalent bond like this. It's actually called a peptide bond. And then dipeptides continue to be added together until you get polypeptides. Poly means many, so this is going to be many that are hooked together. Bear with me for a moment while we draw these because it's important to realize that a single protein might have hundreds of amino acids. Don't worry, I'm not going to make you draw hundreds. And it folds into unique shapes that allow it to serve its function. If anything happens to the folding, the protein won't work anymore. So if the pH is wrong or it gets boiled, then it won't work right anymore. And different parts of the protein might have different folding patterns. When amino acids are linked together to form dipeptides and then polypeptides, it's again this process known as dehydration synthesis. And that means that water is lost each time a covalent bond is formed. So we're going to lose water. Ribosomes are important nanomachine enzymes that link together the amino acids. I'm going to call it an enzymatic nanomachine. It's really quite remarkable. This process is known as protein synthesis, and we're going to look at it more in the chapter on cells. So let's go ahead and put these covalent bonds in between. So I guess it was going this way, was it? There you go. So that would be a full protein. Polypeptides can be broken down back into smaller dipeptides and then to single amino acids. And that process is known as hydrolysis, which means water break. So we're going to have to add water in order to do that breaking. An important enzyme that helps with this process is called pepsin, and it becomes active in the stomach acid and breaks polypeptides down into smaller peptides that can then be acted further upon in the intestine. As I've mentioned, the folding of a polypeptide is critical to its function. We have a structure in our cells called the endoplasmic reticulum. Not all proteins have to go into the endoplasmic reticulum to be folded, but many do. And the structures of a protein are described in four levels. So, first we have the primary structure, and that is just going to be the amino acid names that are in it. And in, of course, the proper order, like the letters of a word, like spelling a word. Then there is the secondary structure, and that's going to be like within the protein. Maybe there's some places where it forms what's called an alpha helix. And then there's some where they form what's called a beta sheet. And then the tertiary structure is all of the entire protein folding. So it's going to include all the alpha helices. And then, believe it or not, there's also something called the quaternary structure. And this is going to be when two or more poly are needed for one functional protein. A couple good examples of that would be the protein hemoglobin that carries oxygen, and also channels in cell membranes that allow things to go in and out. Those are often made from three or four polypeptides that come together to do the one job of making a channel. I'll just add the words here: primary, secondary, and tertiary. Sorry, that got cramped. And then quaternary. Nucleic acids store genetic information, and they also form our cell energy molecule called ATP. The monomer for nucleic acids are nucleotides. Nucleotides are pretty complex in their structure, actually, but all nucleotides will have a phosphate, and all nucleotides will have a 5 carbon sugar, and all nucleotides will have a nitrogenous base. The 5 carbon sugar can either be one that's called deoxyribose, or it can be one that's called ribose. They're very similar looking. And a nucleotide can have one of 5 different nitrogenous bases: thymine, cytosine, guanine, or adenine. So notice that's 4, and then I put thymine in purple. In that case, they either have thymine or uracil, but then the others are the same. Now, these nucleotides can be put together with dehydration synthesis to form and remember, with dehydration synthesis, you're going to lose water as the nucleotides are put together. And then you're going to end up with a polymer known as deoxyribonucleic acid, better known as DNA. And DNA will have a phosphate group, just like all of them in its nucleotides. And then all of its sugars will be the deoxyribose sugar. That's how it gets its name. And it contains thymine rather than uracil for its nitrogenous bases. And of course, it can have cytosine, guanine, or adenine as well. But that's one of the things that makes it different. DNA is double-stranded, like this. It's a double helix. The molecules are huge. You can have millions of nucleotides in one molecule or chromosome. And they have to stay in the nucleus. Dehydration synthesis of nucleotides can also form another nucleic acid called ribonucleic acid, or RNA. Its nucleotides will all have a phosphate, just like you would expect. Their sugar will always be ribose, hence the name. And their nucleotide nitrogenous bases can be cytosine, guanine, adenine, or uracil. So notice that they will not have thymine. Another difference is that RNA is only going to be single-stranded. It's only going to have hundreds of nucleotides, so it's short compared to DNA. And it's able to leave the nucleus and do a lot of jobs in the cell. So it comes in the form of messenger RNA, transfer RNA, and ribosomal RNA, all of which we'll talk about in the cell chapter. Now, there's one more kind of nucleic acid that we should mention, and that's called adenosine triphosphate. It's not a polymer like DNA or RNA. It's a single nucleotide. So put a special structure. Adenosine triphosphate will have adenosine for its nitrogenous base, and that will be hooked to a ribose sugar, and then that will be hooked to 3 phosphates. And if one of these is broken off, energy is released, so it can use this as energy currency in our cells.